So, good evening, everybody, um, and happy Ugadi, Gudi Padwa, and New Year to everybody who is celebrating today. So, we'll start our class now. So, this is the last week class. So, we'll have one more session, maybe on Tuesday for 1 hour, where we'll just try to revise everything and just try to summarize whatever we have learned so far in this course. And that would be it. That would be the end. And then I think you would have your exams. Okay, so in this week, we are going to study about um, epitranscriptome and protein synthesis. Okay, so before starting anything, uh, you should first know what is the meaning of epitranscriptome. Okay, so epitranscriptome includes all the biochemical modifications of the RNA. That means whatever modifications RNA is going through in a cell, all those modifications, all those chemical modifications are known as epitranscriptomes. And these modifications, which are the chemical modification, they affect the RNA transcripts. Okay, it is so epitranscriptomes is all the biological or biochemical or chemical modifications that occurs to the RNA within a cell and which affects the RNA transcript. Now, what is written? That unlike the effects of DNA and histone modifications on gene expression that occur mainly at transcriptional level, the effect of RNA modifications happen at post-transcriptional level. So always remember that whatever effects are happening on RNA modification, it happens at the post-transcriptional levels. And RNA modifications, what all it can affect? It can affect RNA splicing, RNA stability, RNA translation, and as a result RNA modification could upregulate or downregulate gene expression. That means not only DNA modifications can affect the expression of genes, RNA modifications can also upregulate or downregulate the gene expression. Okay, now we'll come to the first lecture, which is the important RNA modifications. That means epitranscriptome is nothing but the RNA modifications that happens post-transcriptional level. So why post-transcriptional level? Because obviously modifications are happening on RNA. That means transcription has already taken place. That means the modifications are post-transcription only, obviously. But all these modifications, they do affect the expression. Okay. So now before uh, uh, reading about the important RNA modifications. So, in the last week's class, we were studying about telomeres and the enzyme telomerase, and we studied various things about telomeres and telomerase. So, here are some of the examples just to show that what telomeres and telomerase can actually do. So, telomerase, can anybody tell me what are telomerase? What is the meaning of telomerase? Telomerase means.
Reverse transcriptase.
Okay, yes. So it is a reverse transcriptase. It's an enzyme which is responsible for the addition of telomeres at the end of the chromosome. Okay, so these, just a minute. Okay. So these telomerase enzymes can immortalize the cells. Immortalize the cells means we have studied that there are different kinds of uh, cell in our body. We have somatic cells, we have cancerous cells, and we have germ cells. And we studied that the telomeres gradually shorten in the case of somatic cells, but in the case of germ cells and in some of the stem cells, the telomeres are maintained at a constant rate because telomerase enzyme is present in germ cells. And in cancerous cells, what happens? Because there is an upregulation of telomerase enzyme, and because there is an upregulation of this telomerase enzyme, the telomeres are constantly getting add up after each division, after each cell cycle, and this leads to the cell become immortalized because it's never going to die. Because one of the reasons that cells eventually die is because the telomeres eventually gets shortened up, and therefore the cells gets exhausted of telomeres and the cell die. But If telomerase enzyme is present, then the cell cannot die, because after every round of replication, new telomeres are added with the help of telomerase enzyme. So, in a way, telomerase can immortalize the cells. Okay, so this is a study which has been published in the journal Science, which says that, I think someone, just a minute, yeah, fine, which says that human retinal pigment epithelial cells and this foreskin fibroblast were transferred with vector encoding the human telomerase catalytic subunit. Okay, and control cells show telomere shortening as well as normal level of beta galactosides as a mark of cellular senescence. But telomerase in transfected cells exhibit longer telomeres and reduced level of beta galactosidase. That means these are the different studies that have been done. It is a study which has been done on retinal pigment epithelial cells, and what they showed and what they could observe was that when they transfected the cells with telomerase enzyme, the telomeres were long and there was reduced level of beta galactosidase enzyme. Okay, so that means telomerase is immortalizing the cells. Telomerase can restore youthful phenotype in live tissue. Again, the logic remains the same. How it can restore the youthful phenotype? Because it is not letting the cells to die. Okay, what happens? Why our cells age? Because telomeres are getting shortened up, and that's why you can see wrinkles on the face, and different kinds of pigmentations and black spots and dark spots on the face, because cells are eventually getting aged, cells are aging, cells are dying. But if you have telomerase in your cells, then your cells are not aging at all, because after every round of your cell division, whatever telomeres are lost, again they are added up by telomerase. Therefore, telomerase, it can restore the youthful phenotype. Now, restore means that suppose an aged person or someone who is in nearly 30s start taking telomerase enzyme, then the aging can be delayed a little bit. You cannot stop aging, but it can be delayed to a certain limit. So again, they did another study in which what they did was normal human dermal fibroblasts were transfected with HTERT. What is HTERT? Here it is written, human telomerase catalytic subunit. Okay. And then what they could observe was that mice grafted with telomerase positive cells exhibited a youthful phenotype despite the same number of replications. Okay. So telomerase can immortalize the cells. Telomerase can restore the youthful phenotype. Or what else it can do? It can restore the youthful phenotype in whole organism. Again, a study that has been done where the restoration of youthful phenotype was observed in testis, spleen, liver, intestine, and also there were some other effects. Not only that, it is, see, everything, each and every cell, each and every organ of our body starts aging. Okay, we all age. And when I talk about aging, it's not about looking youthful, it's not about looking young and pretty, it's also about the proper functioning and the working of your organs. Okay, so telomerase cannot only make you feel you young by reducing the wrinkles and everything on your face, but it can also, up to a certain extent, uh, uh, restore the function of certain organs of our body. For example, it can restore the fertility. There can be reversal of cerebral atrophy, reactivation of neural progenitor cells. That means all these functions can also be achieved by the enzyme telomerase, by the transfection of telomerase. Then telomerase activity predicts longevity. Obviously, as healthy your cell is depends upon as many telomeres you have, and the number of telomeres depends upon whether you have telomerase or not. If telomerase is not there, then cells eventually will die and it will have a it will not survive for a very long number of replication. It all depends upon the number of telomeres you have. Okay, so if high telomerase activity will result in longevity. Fine. Okay. Okay. So up till now, we studied that telomeres can immortalize the cells. Telomeres can restore the youthful phenotype. Telomeres can also restore the youthful phenotype in other different organs, and it can have some other functions as well. And telomeres activity predicts the longevity. Now comes another term, which is telomerase activators. What is the meaning of telomerase activator? So up till now we have studied that telomeres, which are the short repeat sequences which are added at the which are present at the end of the chromosome, which gets shortened after each round of cell division in somatic cells, can be maintained by the enzyme telomerase. Telomerase is a reverse transcriptase, and the function of telomerase is to add telomeres at the ends of the chromosome. These telomerase are present in germ cells, they are present in some of the stem cells, and they are present or they are upregulated in cancerous cells. But in the case of somatic cells, these telomerase are not present. This is what we have studied in a gist. I'm telling you in our last week class, and then telomerase have some examples that we just studied. Now comes a question that can there be a factor, a reagent, a chemical or something that can even increase or enhance the activity of telomerase? Because if the activity of your telomerase is enhanced, then see, always remember that telomerase is not always bad. See, if your telomerase activity is enhanced, it is good for your germ cells. It is good for your fertility. So telomerase cannot cause cancer. Cancerous cells have an upregulation of telomerase. Okay, these two are different things. Cancerous cells have an upregulation of various kinds of metabolic pathways and various other factors, and also telomerase. So telomerase are upregulated in cancerous cells, but telomerase cannot cause cancer. Remember this. Okay. So yes, there are some factors. There is one such molecule which is known as telomerase activator, TA65. Okay. And this molecule can enhance the activity of telomerase. And therefore, this molecule is known as telomerase activator because it is causing the activation of telomerase enzyme. Fine. So this TA65, it is a naturally occurring, highly purified single molecule derived from the Chinese herb astragalus, and it activates the HTERT gene. What is this HTERT gene? Here we have studied. This is the catalytic subunit. So in telomerase, if I'm not making the whole structure of telomerase. So every protein has different subunits. Okay, the structurally, if we see any protein, every protein has different subunits. So the catalytic subunit of telomerase, the catalytic the catalytic subunit of telomerase is actually responsible for the addition of telomeres. So if you want to transfect telomerase to any cells, no need of transfecting the whole telomerase. You just need to transfer the catalytic subunit. This would increase the chances of transfection also, because smaller the molecule, greater the chances of transfection. So what happens that this telomerase activator 65, it activates this catalytic subunit. Okay. And in vitro, it has been shown that it moderately activates the telomerase in human keratinocytes, fibroblasts, and immune cells. And in vivo also, it has been administered, and it has been shown to activate the telomerase activity. So what we could understand from here is that telomerase activity can be enhanced with the help of some telomerase activators. One such example is TA65. Now this, what we are doing, we are artificially providing to ourselves something externally, we are adding up. Okay. Apart from external addition of something, you can also do some intrinsic activities that will enhance the telomerase activity. Fine. And what are those intrinsic activities that one can do that can enhance the telomerase activity? Studies has been shown that exercise is one such thing that can enhance the telomerase activity. So there has been studies which has shown that voluntary running for 3 weeks, it has caused 2 to 9 fold increase in aortic telomerase and 3.3 fold increase in telomerase activity in the mononuclear cells. This is in murine, means in mouse model. In human model, it has also been shown that there is 2 to 5 fold increase in telomerase activity in athletes and 1.8 fold increase in telomerase activity in middle age athletes. That means if you want to enhance your telomerase activity, then you should exercise. So exercise is a remedy, it's a solution of so many problems, including this telomerase activity also. Fine. Now we'll come to the topic that we have. This was all the continuation of what we have studied in the last week. Now we'll come to our topic for the week, which is epitranscriptome. So for those who have joined late, I'll just quickly revise that epitranscriptome is nothing but all the biochemical modifications that happens on the cell, on the RNA. Whatever biological modifications that are happening on the RNA, those all modifications combined together forms the epitranscriptome of the cell. So what we're talking about? We're talking about the various RNA modifications here. Fine. And these RNA modifications, it can affect RNA splicing, RNA stability, RNA translation, everything it can affect. Epitranscriptome. Fine. So now we'll see what kind of RNA modification happens that leads to the formation of epitranscriptome in the cell. So are you guys following up till here? Are things clear? Are you able to understand? Fine. So if you have any doubts or any problems, you can ask me. Even in between, you can just okay, fine. You can just message me and I'll try to resolve your problem. Fine. So now we'll talk about the different RNA modifications that happens that leads to the formation of epitranscriptome in the cell. So one such important RNA modification is N6-methyl adenosine. Okay, this is the important modification that happens on RNA. N6-methyl adenosine. Now it is an abundant modification in the mRNA and in DNA. It is a dynamic and reversible process and most pervasive modification in human mRNA. And this modification, it is present in viruses, mammals, insects, plants, yeast, and it is also found in some of the non-coding RNA such as tRNA, rRNA, nuclear RNA, snRNA. Okay, just a minute. Okay, fine. Then the methylation of adenosine. So what is this modification? It is N6 methyl adenosine. That means at the sixth position of nitrogen, at the sixth position there is a nitrogen where methyl group is added and it becomes methyl adenosine. So what is happening? This modification is nothing but the methylation of adenosine. Okay, this is a, just a minute, people are now adding. This modification is nothing but the methylation of adenosine. So The methylation of adenosine is directed by a large M6 adenosine methyl transferase complex containing. So you just need to understand that one of the important RNA modification that happens is N6 methyl adenosine. That means the methylation of adenosines. And this methylation of adenosine is done with the help of the enzyme M6A methyl transferase complex. This is the complex which causes the methylation of adenosine, which contains METTL3 as the SAM binding subunit. That is just to, these are all just to mug up. You just need to understand the logic. Then what? In vitro, this methyl transferase complex preferentially methylates the RNA oligonucleotides containing GGACU, and similar preference was identified in MAPS. Okay, fine. You just need to read this. So you just need to understand that epitranscriptome means the RNA modifications that happens on the RNA post-transcriptionally, obviously. And an example of one such modification is N6 methyl adenosine. This is the methylation of adenosine which happens in some viruses and eukaryotes like mammals, yeast, plant. That means this methylation is found across all the species, almost all the species. In mammals also, insects also, plants also, yeast also, in viruses also, and in RNAs, in non-coding RNAs such as tRNA, rRNA, and long non-coding RNAs also. So it's a very common RNA modification. Fine, and it happens with the help of this complex, which is M6 methyl transferase complex. Fine. Then, so now come three terminologies that you should understand. One is writer, next is readers, and last one, eraser. What is the meaning of this? So this modification, M6 methyl adenosine, is regulated by three kinds of protein complexes: writers, reader, and eraser. Writer means the one who writes. Reader means reader who reads. Eraser, that erases. So what, in this context, what can be a writer? What can be a writer in this context? Can anybody try to think? What is writer here?
RNA modifications.
Hmm. Okay. Fine. So I'm saying this modification is regulated by writer, readers, eraser. Writer means factors that writes or factors that cause this modification, that adds methyl group to the adenosine molecule. Okay, that adds the methyl group. It is doing the work, it is writing, it is adding the methyl group. Reader means that reads this modification, reads the methyl group. And eraser means that erases this modification. So this is how this modification is regulated. Writer, that adds the methyl group. reader that reads this modification, that reads the methyl group, and eraser that erases the methyl group. This modification is nothing but the methyl group. Are these three terms clear to everybody? Because we will be using these terms again and again in our whole lecture now. Hmm? Okay. So fine. So I assume that these three terminologies are clear to you. Writer, reader, and eraser. Okay. So far we have studied that epitranscriptome is all the RNA modifications that happens on the cell, and one such RNA modification is N6 methyl adenosine. Okay, that means methylation of the adenosine, and this modification is regulated by three kinds of factors: writer, reader, and eraser. Writer are the molecules that acts a methyl group. Reader are the molecule that reads this methyl group, and eraser are the molecule that erases the methyl group. Okay, so what is written over here? So more recent studies have characterized other key components of M6A methyl transferase complex, that is writers. And examples of writers are this: METTL 14, Wilms tumor 1 associated protein. These all are the examples of writers. Okay. The hypothesis is that M6A adenosine in mRNA, being dynamic and reversible, is proven with the discovery of first M6A demethylase, which is FTO. That means these methyl transferase, that means this kind of methylation, this methylation of adenosine is reversible. That means it can be removed also, and it can be removed with the help of erasers. So erasers are nothing but demethylase. Okay, demethylase means which removes the methyl group. So erasers are demethylase, which removes the methyl group. So these modifications are dynamic. That means it can be reversed, and they can be reversed with the help of erasers. One such example is FTO. Another M6A demethylase is ALKBH5, and later they are known as erasers. Then the biological function of M6A are mediated through a group of RNA binding protein that specifically recognize the methylated adenosine on RNA, which is reader, and these binding proteins are named M6A reader. That means, okay, writers are the ones who adds a methyl group. Now, after addition of the methyl group, whatever biological function it has to perform, it has to be read. It has to be recognized. Now, so some molecule should be there that can see that, okay, acha, now this adenosine has been methylated. So now it has to perform some different function. So there has to be some molecule that can see, that can observe, and that can eventually perform such action, that there is methyl group added on the adenosine. So that function is performed by proteins which are known as readers. So readers are the proteins that can recognize the methylated adenosine. So addition of methyl or the modification, this N6 methyl adenosine modification, addition is one thing and biological function is another thing. Addition is done by writers and biological function is performed by readers. And demethylase activity, that means erasing the modification, that is done by readers. Okay. Now we'll just see the question. So first chapter and some last two chapters, those are very important, because last two chapters we have protein translation, and that is important chapter, not just for your exam point of view, but in general that is an important topic for your competitive examinations also. Okay, so first try to solve this question: which of the following is a frequent RNA modification? deadenylation, ubiquitination, N6-methyl adenylation, and sumolation. I'll give you 1 minute and then I'll switch the screen. Okay, I'm switching the screen now. Let me see what you all have answered. So Janara has answered C, Kanchana has answered C, Kavita has answered C. Others, Divya, what do you think should be the answer? Sandhya, Vanaja, what do you think should be the answer? Divya, what do you think? I want little more participation. It's a very easy question, right? Tejaswini has answered C. Fine. Divya has also answered C. Good. Good. That means you're listening. Fine. The correct answer is C. So we are all theory that we are studying, all this story, it is about this N6-methyl adenylation only. So the correct answer is C. Fine. Now we'll come to lecture 2, which is again. So what we are studying, we'll just continue about it. Readers, writers, and erasers. So some examples of readers are YTHDF1, YTHDF2, YTHDF3. So this YTH domain are the examples of reader, and they have a conserved M6A binding pocket. Then others such as insulin growth factor, they are also some class of reader. So these are all the examples of readers. Okay, these are all the examples of readers that you need to learn. The YTH domain, then insulin growth factor. Now you can ask that insulin growth factor, ma'am, insulin growth factor, that is a different, it has a different function. How it can recognize the M6A modifications also? So in biology, in cell, what happens that one protein can have more than one functions. One function would be its primary function, and other function would be its secondary function, which it shows at certain stages or in certain conditions. And very good example is here only, that's insulin growth factor. So insulin growth factor is has a different function, but it can also act as a reader in some of the cases in context dependent manner. Fine. Then these M6A readers together with the writers and erasers establish a complex mechanism of M6A regulation, in which writers and eraser determine the distribution of M6A on RNA, whereas readers mediate M6A dependent functions. And M6A has also been shown to mediate a structural switch termed MCA switch. So what does it mean? So basically these writers, readers, and erasers, these are nothing but the proteins that regulate the function of this modification. So distribution of this modification or RNA would be controlled by writer and eraser, because writer will write the modifications, eraser will erase the modification. So distribution, that as and when where it is present in the whole RNA, that is decided by writers and eraser. Okay, and the function, the M6A dependent function, that is mediated by reader. So biological relevance of this modification is attributed because of readers. Whereas what the places where this writers and eraser would be, where it is present, that is regulated or determined by writers and eraser. Is this clear to everybody? The difference in the working of writer, eraser, and reader? What are the different functions of writer, eraser, and readers? Is it clear? Vanija, is it clear to you? If it is clear, just thumbs up, or if it is not clear, you can ask me. Okay. Okay, once more. Okay, Prishila, what is your question? You can also ask me. Okay, so I'll just explain it once more. Okay, see, suppose this is the RNA molecule, and just assume for our convenience, this is the adenosine. Just assume it, okay? And here is the modification. M6A. Okay. Now, all the places, these are the places where adenosine is present. And in all these places, this, sorry, this modification is there. M6A, 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 M6A. So the addition of these modification is done by writers. Just like we write on the copy, just like I am writing, I am adding something on this particular white screen. Similarly, writer proteins will come and they will write the modifications on the adenosine moieties which are present on the RNA. Okay, now erasers will come. So what will happen? This is what the function of writers. Again, RNA molecule. Again, these modifications. Again, I'm just writing here. M6A, 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 and M6A. So these are the modifications that has been written or added by writers. Now erasers will come and say, I don't want a modification here. I don't want a modification here. So now earlier we had 1, 2, 3, 4, 5 modifications. Now we have only 1, 2, 3 modifications. So this removal of the modification is done by demethylase, which demethylates. So writers are methyl transferase, which adds the methyl group, and eraser eraser are demethylase. D means removing, that removes the methyl group. So now eraser has removed the methyl group. So from this, what we can say? We can say that from here it can be said that the distribution means the places where this modification would be present. This distribution is mediated by the combined action of writers and eraser. Distribution means where the places, wherever, whatever places the modification would be present, that is determined by writers and eraser. Is this clear? Up till now, is it clear? Are you able to follow the difference between writers and eraser?
Yes, ma'am.
Okay. Now comes readers. Again, I'll make this. This is your RNA molecule. These are the adenosine moieties. Just for your um, visualization, I'm doing it like this. And these are the places where modification has been actually done. Okay. Now, okay. Writers have come, they have added the modifications. Eraser came, they removed the modifications from places they did not wanted. Now what? Now what is the next step? These modifications are present. Now these modifications should perform their function, should perform their biological function, whatever function they have to perform. Okay, so that means, for example, if you have a protein or a ligand, this ligand can perform its activity only when a receptor is there which can receive it, and ultimately the signaling will go. Any protein molecule, any ligand can perform its activity only when it is bound to a receptor. So there has to be a receptor which will recognize this ligand, and ultimately it will start the signaling process. Okay. Likewise, here also, consider these modifications as ligands. Now these modifications has to be recognized by some proteins, and ultimately after the recognition, function would be performed. So that recognition would be performed by whom?
readers,
Readers. Now readers will come. These are readers which will read the modifications. Here comes reader. It read the modification. So the distribution of the modification is determined by writer and eraser, and the biological relevance would be determined by reader. Now is it clear to everybody? I have tried to explain in the most simplest way. Okay, fine. So there was one more girl. I don't, just a minute. So Prishila, is it clear to you now? You had raised your hand. So is it clear to you? Okay, I'm assuming. Yeah. Okay, fine. Good. So we have to move a little fast because we are still on the second chapter, but I wanted you guys to understand it clearly, because other things you can just read out. I mean, that's not difficult. But if you, once you know the concept, if you have built the concept, then you can just read the details. That is not difficult. Okay. So that is what it is written over here. Now, what they are saying, that the differential M6A modification profiling is an emerging hallmark of cancer. Why? Because aberrant M6A deposition, it plays a key role in tumorigenesis in various cancers by altering the gene expression. Fine. Then M6A methylation plays an important role in wide variety of biological and pathological process, okay, including cancer development. Fine. Then this particular methyl transferase is upregulated in this cancer, okay. Now this M6A modification frequently invoke key components of oncogenic pathways. That means this kind of modification, this M6A modification, it can ultimately lead to cancerous condition by altering some of the pathways such as Wnt pathway and PI3K pathway. Okay. So, understanding the function and molecular mechanism of M6A mediated epitranscriptome may thus unravel novel therapeutic targets and biomarkers for cancer detection. That is all fine. So, you can see over here how this. So, what is this YTDH1? This is a writer or eraser or a reader. We have studied just now. Think, what is this YTH? Hmm. This YTH is reader. Just read this and these sentences. Hmm. Read. So this YTH are the readers. Okay. Hmm. Now you can see that this YTH is a sorry. Hmm. This YTH is a reader, and this can ultimately, so when YTH reads the methyl 6 modification, it can ultimately lead to tumor genesis and metastasis, stemness, drug resistance, autophagy, bacterial infection, immunity by different pathways. So this YTH reader, when it reads the methyl adenosine modification, it can cause the upregulation of these pathways in different cells. It can cause the upregulation of Wnt beta catenyl pathway, which can lead to tumor genesis and metastasis. It can cause the upregulation of TGF beta pathway. It can cause the upregulation of JAK-STAT pathway, which can ultimately show cancerous phenotypes. Okay, nothing much to understand and read. Just you need to understand that this M6A modification is involved in many kinds of diseases, including cancer. And when this M6A modification is read by reader molecules, specifically YTHDF1, this molecule, this fact protein, it can lead to different kinds of cancerous conditions in different cells. Okay, this is only you have to understand. Now, let us try to solve these two questions. Huh. Factors that cause an RNA modification is called reader, writers, erasers, or modifiers. Think about it. I'll give you 1 minute. Hmm. Okay, now I'm switching the screen. Kavita has answered reader. Tejaswini has answered reader. Then Kavita has again answered D, modifiers. Jahanara has answered B, which is writers. Okay. I want more participation. We have very different varieties of answers. Hmm. See, there are three terms: factors that cause the RNA modification, that erases the RNA modification, and that reads the RNA modification. It's a hint. Now, tell me what should be the answer. There are three terms: cause, erase, and read. So, Kavita has answered writers, Janara has answered B, and Tejaswini. Yeah. So, the correct answer would be B. That is writers. So, writer, they writes the modification. That means they are causing the modification. Readers will read the modification. Readers will not cause the modification. They will just read the modification. And eraser is simple. It will erase the modification. Okay. Remember it. Cause, writes means writing, erasing means removing, reader means reading, which leads to biological relevance. Okay, next question is YTH domain proteins are considered as RNA modifiers, epitranscriptome writers, epitranscriptome readers, epitranscriptome suppressors. Think. I will switch the screen after 1 minute. Hmm. Okay, I'm switching the screen now. Hmm. So Janara has answered C, Kavita has answered C, Prashila has answered C. Trishala, you want to try? Okay, fine. So now I think everybody has understood it. The correct answer is reader. We have discussed it also here. This YTH domain, which is a reader. Fine, good. Now, now we have read that okay, this is the kind of RNA modification that happens on RNA. That is methylation of the adenosine. And this biological implication is mediated by writer, reader, or eraser. Tell me, biological function is implicated by writer, reader, or eraser. What, how biological relevance of the modification is regulated or achieved by writers, by readers, or by erasers. Readers. Good. So readers, because when they read the modifications, they ultimately transmit it. So therefore, readers are the ones that regulate the biological implication of the RNA modifications. So here you can see we have just studied that the biological implication of this RNA modification, one very known biological implication of this RNA modification is cancer. So you can see here they have explained with the help of this Wnt pathway. So what happens is, see, first I'll try to briefly explain you this Wnt pathway. Hmm. Um, huh. See, so Wnt pathway start by Wnt protein coming and binding to its receptor, frizzled, and LRP. It can have various variants 5, 7, 9. LRP can have very variants 5 or 6. So the frizzled and LRP6 are the two receptors. So Wnt comes and it binds. So this leads to the production of another protein which is disheveled, and this disheveled protein inhibit the activity of GSK3 beta. So normally, if Wnt is not there, if Wnt ligand is not there, what happens? Then GSK3 beta, it degrades the beta catenin, and there is no beta catenin now. How it degrades? Because this GSK3 beta, it phosphorylates the beta catenin, and after the beta catenin is phosphorylated, it is marked for ubiquitination and it is degraded. But if Wnt is present, then what happens? Wnt will produce disheveled, and this disheveled will inhibit the function of GSK3 beta. Now, since the function of GSK3 beta is inhibited, there is no degradation of the beta catenin, and therefore beta catenin can now happily go to the nucleus. In the nucleus, it will bind to TCF4 and it will cause the transcription of Wnt target genes. Okay, and one such Wnt target gene is MYC, c-MYC. There are many Wnt target genes. One example of Wnt target gene is c-MYC. And this c-MYC, so what happens? The upregulation of Wnt target genes, it causes increased translation of this YTDHF1. So there is increase translation of YTHDF1. And this increased translation of YTTHDF1, what it does? Now, there is increased production of the receptors. And when there is increased production of receptors, more and more receptors are present, more and more receptors are there to bind with the Wnt. So ultimately what is happening, if this modification is present, M6A modification is present, then reader will come and it will read the modification. That means reader comes only when this modification is present. Now what happens, that this Wnt signaling will go, it will produce the target genes. I mean, it will transcribe the target genes. These target genes will cause more and more translation of this YTH1, which will again cause more and more signaling. That means it is acting as a positive loop. Wind produces target genes increases YTDH1, and this YTDH1 ultimately increases the Wnt pathway. So there is a positive regulation. So that means if this modification is present, then what will happen? This modification will cause increase production of frizzled and LRP5, and therefore more receptors are there, more receptor to join Wnt. So Wnt pathway is upregulated. So ultimately the gist of this modification is that Wnt pathway is upregulated because of this M6A modification. Is it clear to everybody how this modification is causing the upregulation of this pathway? Is it clear, or you have any doubts? Okay, fine. So just you have to understand that we have, and if there is upregulation of this pathway produces target genes which are pro-mitotic. That means that promotes the proliferation. And when proliferation, it can ultimately leads to increased cancerous conditions. So this is how everything is related. We are saying we are trying to understand the biological implications of this modification. So the biological implication of this modification is that this modification causes the upregulation of Wnt pathway, and the Wnt pathway ultimately produces some pro-mitotic genes which are involved in proliferation. Therefore, as this pathway is upregulated, it can enhance the cancerous conditions. Okay, fine. So I hope it is clear to you. If it is not clear, you can always go and see the recordings. We have to cover the rest of the part also. Therefore, I'm going to the next topic. And this is all you can just, this is I have kept here just for your reading. There is nothing much to explain over here. You just have to understand that this M6A, so we are studying the biological relevance. Na? We are studying the biological importance of M6A modification. So, this M6A modification can lead to elongation of the circadian period. Then, if this MTTE3 is overexpressed, it can lead to shorter period of circadian clock. Then, these are all just the various biological phenomena that are affected by these M6A modifications. Okay, you can just read it over here. Nothing much to explain. All the thing that was a little logical, I already explained it. Okay, so now up till here, we have studied about epitranscriptome. Epitranscriptome means, what is the meaning of epitranscriptome? Tell me, what is the meaning of epitranscriptome? First thing we discussed today, epitranscriptome. What is the meaning of epitranscriptome?
Biochemical modifications of
Yes,
RNA.
biochemical modifications on RNA. So, what all the biochemical modifications that occurs on RNA combined together, all those what biological modifications are known as epitranscriptome. We studied about M6 methyl transfer, M6 methyl adenosine, which is a RNA modification. Then how it is regulated? It is regulated by writer, readers, and erasers. That we have studied. Then we studied about um, what is the function of writer, eraser, and reader. And then at last we saw the biological implication of these modifications. Now we are switching to the next part of today's lecture, that is protein synthesis. Epitranscriptome we have covered. Now we'll see the protein synthesis. Fine. So protein synthesis, Pratitla, we are talking about translation. So because our course is RNA biology, so we'll try to see the role of RNA in protein translation because we are studying about RNA biology. Up till now, we studied about RNA modifications. Now we'll see protein translation, the machinery, the mechanism also, because mechanism is also very important for you to understand. We have already studied the transcription, we have covered post-transcriptional modifications also we have covered. So we have to cover this protein translation also. But first, we'll see the role of RNA in this translation. Okay. So, what is the role of RNA? There are three roles of RNA in protein translation. Okay. So, we have mRNA, rRNA, and tRNA. All the three RNAs are responsible for the proper translation process to occur. How? Because mRNA has the triplet codon that will specify which amino acid has to come. So mRNA specifies amino acid sequence. That means when the cell is making proteins, when cell is making protein stretch, which protein has to be added, that is decided by the sequence of the mRNA. So mRNA will decide which protein has to be added. So mRNA specifies the amino acid sequence. Each amino acid is this is the function of mRNA. Then tRNA, then whatever amino acid that has been selected by mRNA, it is brought to the ribosome with the help of tRNA. So, what we can say is that mRNA selects the amino acid, tRNA brings the amino acid. And what is the function of rRNA? We'll just see. So tRNA converts the information in mRNA codon into amino acid sequence. So tRNA carry the amino acid specified by the codons and base pair the codons with their anticodons. Okay. Then ribosomal RNA, they are present in ribosomes, which are the translation factory, and specifically ribozyme, which is a catalytic kind of rRNA. It causes the peptide bond formation. So this is all the function of RNA molecules in translation. mRNA, it selects the amino acid because the triplet codon, it specifies which amino acid has to be added. tRNA, it brings that amino acid. RNA is a component of the ribosomes, which is involved in protein synthesis. Also, we have ribozymes, and these ribozymes are nothing but the enzymes that causes the peptide bond formation. Now, next is the genetic code. You can see over here. See, this is the genetic code. Okay, I'll just do it like this. It will be better. Hmm. Hmm. So you can see this is a genetic code. We have 61 codons that specify the amino acid. This we have already studied. So three uh, codons, three ATGC, three codons makes one amino acid. So first position, second position, third position. That means U, U, U. How do we read it? U, U, U. U, U, C. U, U, A, U, U, G. So this is the first position. This is the second position. This is the third position. And there is another way by which you can read it this. How you can read this? So there is one thing that you should know, that one amino acid can be coded by more than one codon. What I'm saying? I am saying, just a minute. I am saying that, oh, sorry. I am saying that one amino acid, it can be coded by more than one codon. So if I say alanine, mm, alanine. So alanine can be coded by one, two, three, four different kinds of codons. And the difference in the different kinds of codons is at the third position. So if alanine is GCU, GCU. So this is only different in different kinds of codons of alanine, and this is known as wobble base. Wobble base means that it can change. See, if I talk about alanine, GCU, GCC, GCA, GCG, change is always at the third position. Always remember this, very important. Third place is known as a wobble base. Okay, then so we have 61 codons that specify the amino acids. There are three stop codons which are known as the termination codon. Normally, the first codon is AUG, which specifies methionine, which serves as a start codon. So, in general, your start codon is methionine. Okay, fine. Then, how these triplets can be read? So, in frame, it has to be read as 3, 3, 3 because 3 codons specifies one amino acid. So, are you guys following this? Are you guys able to follow this?
Yes, ma'am.
Okay. So, three codons specifies one amino acid. So, you can see how you can read it. So you can read it as 1 2 3 1 2 3 like this. This is your frame 1. Or you can read it leaving the first one 1 2 3 1 2 3. This is the frame 2. Or leaving the 2, then this is the frame 3. So in these three frames your triplet can be read. Now important thing is that what will decide that which frame is to be used. It depends upon in which frame you are getting your AUG start codon. Okay, if you get your AUG start codon in frame 1 would be used. If you get your AUG start codon in frame 2 would be used. And if you get your AUG start codon in frame 3, then frame 3 would be used. So which frame has to be used? It depends upon that in which frame you are getting the start codon AUG. Is it clear to everybody? I hope it is clear. Fine. Okay. Then how mRNA is decoded. So I think this all you know. So what happens? First, charging of tRNA takes place. What is the meaning of charging of tRNA? So charging of tRNA means that whatever amino acid has to be brought in, that acid has to be attached to the tRNA. And how it is attached? It is a two-step process. So amino acids are attached in ester linkage through the to the 3' terminus of the tRNA, forming amino acyl tRNA. That means the tRNA has to be charged. That means whatever amino acid it has to bring, it has to be added. So this is done with the help of enzyme amino acyl tRNA synthesis. Okay, and this is a two-step process. First, what happens? That you see, this is the tRNA and this is the amino acid. So first what happens, that this amino acid, which is phenylalanine, it gets linked to the tRNA. It forms amino acyl tRNA. Now this amino acyl tRNA binds with the codon, the mRNA codon. And this is how the two-step processing for mRNA decoding happens. So first, tRNA is charged by the addition of the amino acid, and then in step 2, that tRNA is added to the growing chain by codon-anticodon interaction. Is it clear? If this is your...
Ma'am, repeat once, ma'am.
Sorry?
Repeat once, ma'am.
Okay. See, first thing is charging of the tRNA. What does it means? Charging means addition of the amino acid that has to be added to the growing chain. So if this is your tRNA, I'm just making it, it's not a very good tRNA. At the 3' end, your amino acid is added. Fine. So this addition of amino acid to the 3' end of the tRNA, this is known as charging of the tRNA. And this charging of the tRNA requires ATP. And once this tRNA, it has the amino acid, then this is known as a charged tRNA. And this charged tRNA is added to the growing chain. That is what is written over here. Amino acids are attached in ester linkage to the 3' end of the tRNA, forming amino acyl tRNA. The enzymes that carry out this ATP driven reaction are known as amino acyl tRNA synthetase. So normal tRNA, this is a tRNA. In this tRNA plus the amino acid is added. And this tRNA, which is a charged tRNA, it is known as amino acyl tRNA. This requires ATP and the enzyme is amino acyl tRNA synthetase. This is your charging of tRNA. This is the first step. The first step in protein synthesis is the charging of tRNA, which means the amino acid is attached to the 3' end of the tRNA. This addition requires ATP and the enzyme that is required is amino acyl tRNA synthesis. So now your amino tRNA is charged, means amino acid is added. Amino acid added to the tRNA. This is your first step. Then what would be your second step? Second step is this tRNA which has the amino acid, it is added to the growing polypeptide chain. And this is what the two-step process they are saying. Okay. So this is the step one. Step one. This is your amino acyl tRNA. Now what happens? In step 2, the amino acid is added to the growing chain based on codon-anticodon interaction between mRNA and tRNA. So basically now this is added to the chain based on the interactions that we'll see later. Just for now, you just understand that first, see, the function of tRNA is to bring the amino acid. So first the amino acid has to be attached to the tRNA, and that attachment of the amino acid to the tRNA is known as charging of the tRNA. Then that tRNA adds the amino acid based on the codon-anticodon interactions. That means that codon is present in the mRNA and anticodon is present in the tRNA. It is just like how A binds with T, G binds with C. So like this only, what happens? See, this is the anticodon. Actually, this tRNA has to bind with the mRNA, no? So how it will bind? See, this is the anticodon, which is AAA. And this is the codon, which is UUU. So there is an interaction between codon and anticodon, just like complementarity is there. Okay. So the net result is that this is added to the growing chain. Now is it clear?
Yes, ma'am.
Fine. Then this is the structure of tRNA. Nothing much to explain. So this is the secondary structure and this is the tertiary structure. This you need to understand. Secondary structure is a clover leaf structure and tertiary structure is a L-shaped structure. This is your L-shaped structure. Like this. And this is your clover leaf structure. So you just need to understand that this is your secondary structure. This is your tertiary structure. And you can see for your understanding perspective, secondary structure is important. See, this is your anticodon loop, which base pairs with the codon of the mRNA. This is the acceptor arm because it accepts the amino acid. This is a 3' arm. So this is just in a reverse way it is added here. This is acceptor arm This is the acceptor arm. So acceptor arm at the end has CCA. So always remember that the amino acid, whatever amino acid is coming, it is added to the 3' end of the tRNA. And the 3' end of the tRNA, whether it is present left side, right side, that 3' end of the tRNA is known as acceptor arm. And that acceptor arm ends with CCA. This you have to remember. Then the amino acid is attached in ester linkage and many residues are modified in tRNA. That is okay. Just you need to understand in the structure of tRNA that secondary structure is a clover leaf structure. Clover leaf secondary structure. An L-shaped tertiary structure. There is an anticodon loop that contains the anticodon, and there is an acceptor arm that ends with CCA. Okay, this is your tRNA structure. Then codon anticodon base pairing. See, now pay attention here. This is anticodon and this is codon. This anticodon is read like 1, 2, 3. Codon is read like 1, 2, 3. Why? Because you always read from 5' to 3' direction. So here the 5' to 3' direction is like this. So 1, 2, 3, 1, 2, 3. So 3 will pair with 1, 2 will pair with 2 and 1 will pair with 3. I mean, this is just the position, whatever it is there. So there is hydrogen bonding. Just like we saw here, there is hydrogen bonding between anticodon and codon. Okay. So hydrogen bonding between the first and second position of the codon. You can see first and second position of the codon. This is the first position, this is second position, and third and second position of the anticodon. Third and second position of the anticodon. However, base pairing between the third position of the codon, this is the third position, and the first position of the anticodon is a wobble position. That means these two base pairing is very strict. At third position, differences can be there. And that is why we saw that one amino acid can be coded by more than one codons, and that difference in the codon comes at the third position, which is known as a wobble base. So, just for your explanation, I am making this. This is your tRNA, okay, and this is your mRNA, okay. One, two, three, 3, 2, 1. Why it is like this? Because this is the 5' end, this is the 3' end. So the code we will start reading from the 5' end. And here, this is the 5' end, this is the 3' end. Okay. Now, suppose there is A, so here would be U. C, so here would be G. यहाँ पे if it is G, it would be C. Now what they are saying that these two base pairing is strict and this base pairing it can be changed and it is known as wobble base. And this, because it is a little flexible, this is flexible. That is why here we saw that one amino acid can be coded by more than one codons. See, alanine can be coded by GCU, GCC, GCA, GCG. So change is always in the third position of the codon. Here you can see change is always in the third position of the codon and first position of the anticodon. Is this clear to everybody now?
Yes, ma'am.
Okay, fine. Now, so up till now, we have studied about the charging of the tRNA. Charging of the tRNA means the addition of the amino acid to the tRNA that occurs with the help of the enzyme amino-acyl transferase. So, amino-acyl transferase synthetase, and it requires ATP. And then we studied about the structure of the tRNA. So, structure of the tRNA, we have a secondary structure, which is a clover leaf structure, and a tertiary structure, which is an L-shaped structure. Always remember that there is an anticodon loop which base pairs with the codon, and at the 3' end, whether the 3' end is present at the left side or right side, amino acid always binds at the 3' end, and 3' end ends with CCA. Then we saw the codon anticodon base pairing, and then this is the ribosome composition. So ribosome composition we have been studying, uh, we have studied it many times. We know that there are two types of ribosomes, prokaryotic ribosomes and eukaryotic ribosomes. You can see here, prokaryotic ribosomes are 60S ribosomes, eukaryotic ribosomes are 80S ribosomes. So these are the different subunits that has. Prokaryotic ribosomes have 50S and 30S subunits. Eukaryotic ribosomes have 60S and 40S subunits. And ultimately, they form a 70S and 80S ribosome. Okay. So this is what the ribosome composition. So now we'll start with So this ends the chapter number 1. Now, chapter number 4. Now we'll move to lecture number 5, the last lecture. So up till now, we have studied about all the basic things that are needed for the protein to translate. We need a charged tRNA. That means a tRNA that has the amino acid. We studied what are the different shapes of tRNA, what different types of ribosomes, how tRNA pairs with the mRNA, that means codon, anticodon, base pairing, all these things we have studied. Now, few important things, I'll just tell you like a story. You try to understand and then we'll move to the lectures. Okay. So, once before the translation starts, always remember that the smaller subunit and the larger subunits are not present together. That means smaller subunit and larger subunits are not together binded to each other before translation. Only when the translation has to happen, the larger subunit comes and binds to the smaller subunits. That means smaller subunit of ribosome and larger subunit of ribosomes are not always bound to each other. They are present separately. So this is your mRNA. mRNA is bound with the smaller subunit, and then the larger subunit comes and binds here. One thing. Second thing is that there are three sites, A, P and E. E, P or A. Always remember this. Okay. So exit site, peptidyl transferase site and acceptor site. So what happens? First tRNA that comes. So what is the first tRNA? What it will bring? It will bring methionine, which is AUG. So first amino acid that is added is mostly methionine, which is the start codon. So the first tRNA, that means the first amino acid, is always brought to the P site. Okay. And later on, the amino acid starts coming to A site. Only first amino acid, which is brought by the tRNA, comes to the P side, and later all the other amino acids are brought to the A side. Is it clear what I'm trying to tell? first amino acid comes to the P site and subsequent amino acid comes to the A site. Okay. Now, this tRNA will bring amino acid to the A site. That amino acid again will bind to this existing amino acid chain, and in this way it keeps on increasing. So this means first methionine was there, which came to the P site, then tryptophan came, then phenylalanine would be added here, like this. This is the anticodon, this is the codon, this is the smaller subunit, this is the larger subunit, this is the tRNA which has the anticodon, and this is the amino acid. Just a basic. Now we'll start. So here we're talking about translation initiation in eukaryotes. So in eukaryotes, we have two subunits, 40 subunit and 60S subunit. 40S subunit is the smaller subunit and 60S subunit is the larger subunit, okay, which ultimately makes an 80S subunit. So I have to move a little quickly because we don't have much time. So we'll try how many questions we can solve. Okay, so first, that's why I will move a little quick. If you have any questions, next week also we don't have any topic to cover. We'll just revise everything. So if you are not understanding any topic, we can cover that. Revise it in the next class also. Now, in eukaryotes, what happens? Actually, the diagram that sir has given is very small, but I had to take this diagram only. Anyways, okay. So what happens? The smaller subunit is bound to some factors such as eukaryotic EIF1, EIF3, EIF4. Okay. So what is there? This is the smaller subunit, and this smaller subunit is bind to some factors, and this smaller subunit is bind to the mRNA. So first what happens? These factor comes and binds to the smaller subunit. After that, when this factor comes and bind to the smaller subunit, then what happens? The larger subunit comes and it binds to it. After the larger subunit comes and bind to it, what happens? GTP will bring the charged tRNA. So always remember what is there. First, you have a smaller subunit which is attached to the mRNA. These factors will come and bind to the smaller subunit. As this factor comes and bind to the smaller subunit, it gives a direction, it gives a signal for the larger subunit to come and bind. So then the larger subunit will come and bind. Then what will happen? As soon as the larger subunit is bind, your charged tRNA is brought by this factor EIF2 GTP. It will bring the tRNA. Then what will happen? After the tRNA has come, mRNA now is circularized. This is the linear mRNA which is bound to the smaller subunit of the ribosome. Some initiation factors come and bind to it. When the initiation factors comes and bind to it, larger subunit also comes, which has E, P and A. Now, tRNA comes with the help of some other factor and GTP. You can see here, factor name you can remember, EIF2 GTP. So this tRNA comes now. Now this tRNA has to attach at the P site because this is the first tRNA. First tRNA always attaches to the P site. So then what happens? This tRNA has to attach to the P site. The tRNA attaches to the P site and this linear mRNA is converted into a circular mRNA. Is it clear to everybody? the steps of initiation. This is what initiation happens. And then when this tRNA attaches to the P site, formation of first peptide bonds take place. And then the tRNA would be emptied, and then that tRNA moves to the E site. So what happens is EPA. Quickly, I have to explain because I have to cover the questions also. So first tRNA comes to the P site and methionine is added. So now tRNA does not has any amino acid. That tRNA will move to A site. So this is the sequence. Sorry. I have written it wrong. E, P and A. So first tRNA comes to the P site. It attaches AUG. Now the tRNA is empty. It will go to the exit. This is exit site. Then again tRNA comes to the A site. It attaches the codon here, the amino acid here. It moves to from P to A. So like this, it keeps on moving. Like this to like this. It will go from P it will exit. It will go out. Okay. This is the initiation. So initiation means the coming of the first amino acid and the formation of first peptide bond. That is your initiation. Okay. I think I have. So this is your initiation. And one thing important is that, yeah, okay, for today this much is fine. In detail I will explain you in the next class. I will explain in detail, but just for your reference for your this week assignment per se, I am explaining you this is what happens in initiation. That smaller subunit which is bound by mRNA, what happens, initiation factors bind to it, and then your tRNA comes, and then tRNA comes, and then there are different kinds of proteins are there, and then the tRNA, when it comes, it will bind to the P site and first peptide bond formation takes place. Then we come to the elongation. Yeah, they have explained the initiation in 3 parts, but we will stick to what we have studied just now. Okay, this is what they have saying. EPA that we have already covered. Then comes elongation. Now you see here, what is elongation? What is happening? See, this is your E site. This is your P site and this is your A site. So now the tRNA will bring the amino acid to the A site. This will add to the P, the growing, this is the growing peptide chain. This amino acid will form the peptide bond like this, and then the tRNA would be emptied and it will go to the E side and then it is removed. So like this, it will keeps on adding the amino acid to the growing polypeptide chain. All the new amino acids with the help of tRNA will come first to the A side, then it will form a peptide bond formation, then it will get exit. And like this, the ribosome will keep on moving. It will move like this. Actually, the ribosome is moving, not the mRNA. So this is what you have to understand. This is your elongation. And then comes your termination. What happens in termination? I cannot do it such a big. Mm, You can see here incoming charged tRNA in the A site of the ribosome. So these many ribosomes, these many amino acids have been added. Then peptide bond joins amino acid 3 and 4. This was 3 is here in the P site. 4 also forms a peptide bond here. This is the peptide bond. This is the peptide bond that is formed. Then tRNA3 is released from the ribosome. The A site is ready. Then what happens? See here. This is the tRNA3. This is the tRNA4. The tRNA3 has given this third amino acid, and tRNA4 has brought this fourth amino acid. This fourth amino acid will come to the A site. There is a peptide bond formation. This third tRNA move to the E site and it is released. And this fourth tRNA now moves to the P site. Again, fifth tRNA will come. It will form a peptide bond formation. This fourth will move to the E site and fifth will come to the P site. So like this, the tRNA keeps on shifting. Okay. So now how the termination happens? When a stop codon enters the A site, it is recognized and it is bound by release factor. Now what happens? Here we have studied that incoming amino acids are coming and they are binding to the A site. Now what happens? How this termination is stopped is that despite some stop codon comes and it binds to the A site. That means now there is no amino acid that has come and bounded to the A site, but a stop codon has come and it has bound to the A site. And then when a stop codon comes, then a release factor is initiated and it gets a signal that it has to terminate the transcription. Therefore, transcription is terminated. Okay. So now we'll move to the questions. So what I'm planning is that in the next class, quickly I will explain you the translation, initiation, elongation, and termination and other. So we'll just revise whatever we have studied up till now. And also I will explain you this initiation, elongation, termination once again if you have any queries and problems. Because today we are running out of time, so I cannot give more time for this. But this is a very important topic. Therefore, I will explain you again. So do not panic. If you have not understood anything, we'll again discuss this in the next Tuesday session, because now we don't have anything extra to discuss. Okay. So quickly we'll try to solve some questions now. So mRNA codons are usually discontinuous manner, distinct manner, overlapping manner, or three frame manner. Think. Kanchana has answered D. Janara has also answered 3D. Okay. Now, this is a very confusing question. Why? Because, see, codons are in a three-frame manner. It is correct only. Yes, codons are in a three-frame manner. But what is more important? See, in MCQs, you have to choose the most relevant one. Here, the most relevant one is the B, that is distinct. Distinct means what? I said that one amino acid can be coded by more than one codon. One amino acid can have more than one codon. But can one codon code for more than one amino acid? Tell me. Can one codon code for more than one amino acid? Yes or no? Can AUG codes for methionine? Can AUG code for any other amino acid also? No.
No.
So codons are very distinct in their function. One codon is specific for one amino acid only. One amino acid can be coded by more than one codons, but one codon can only code for one amino acid. And this is the most important property of codons. And therefore, these mRNA codons are distinct. That means if any allele is there, allele can be coded by some, I'm just writing it like, for example, GGU. Just I'm writing, it is not the actual codon. So allele can be coded by GGU, GGG, GC, G, GCA, but GCU cannot code for any other amino acid. It can only code for alanine. This is a very important difference. Okay. So the correct answer is B, that is distinct manner. Then next is, before participation in protein synthesis, the tRNA must get modified, denatured, amino acylated, or trans acylated. Think. C, Kanchana has answered C, Kavita has answered C, Janara has answered C, which is amino acylated. Yes, correct. So, tRNA must be amino acylated. That means amino acid should be added to the tRNA, which is known as the charging of the tRNA. Okay. Next question is, bacterial mRNA codons are not similar to the plant codons, not similar to the virus codon, similar to other animal codons, similar to mitochondrial codons. Try to attempt this. Think what can be the answer. Jayara has answered D. Any other answers? Okay, fine. So, bacterial mRNA codons are similar to other animal codons, because suppose AUG codes for methionine in bacteria also, AUG codes for methionine in animals also. That is why when we study translation in prokaryotes and translation in eukaryotes. So the translation mechanism in prokaryotes and eukaryotes is usually the same only. There are certain differences, and eukaryotic translation is a little more complex than prokaryotic translation, but overall it is the same. The initiation, elongation, termination, the basic logic remains the same, and so the codons also remains the same only, similar only. Then protein synthesis can happen only from mRNA. True or false? Think. Try to answer this question. False. Kavita has answered false. Any other? Tejaswini has answered false. So why you think it is false? See, protein synthesis can happen only from mRNA. So, see, other RNAs also help in protein synthesis. That is true. tRNA also has a role. rRNA also has a role. But there is a reason why tRNA and rRNA are known as non-coding RNA, whereas mRNA is known as coding RNA. Because mRNA can code for proteins and tRNA and RNA cannot code for proteins. That means protein synthesis, the basic prerequisite thing for protein synthesis is that you should have the codes that will specify which amino acid has to be added. If you do not have the amino acid, then what is the uh, purpose of tRNA? tRNA will bring the amino acids. No, but at least you should have first thing that you need is the amino acid sequence. That is the first basic thing. So protein synthesis can happen only from mRNA. Other types of RNA, they will help in the protein synthesis, but the code will be given only from mRNA, and that is why mRNA is known as a coding RNA and other are known as a non-coding RNA. Okay, so answer is true. Next question is,
Ma'am, this question answer, ma'am.
sorry, this question.
mRNA, this is the first
Huh,
question answer.
first question means this bacterial mRNA codons.
Bacterial mRNA codons.
You are asking about the first question, bacterial
Yes,
mRNA codons.
yes, ma'am.
Huh, okay. So, see, whenever I don't know how much you have gone through, but if you study about translation, there is always a comparison of translation between bacterial translation and animal eukaryotic translation, specifically animal translation. Not only translation, whether we are studying about replication, whether we are studying about transcription, we always study two types of transcriptions: prokaryotic, eukaryotic. Translation, prokaryotic, eukaryotic. We never study about plant and animal translation, or virus or animal translation, or bacteria or virus translation. Why? Because over the years, through research, it has been shown that whether it is transcription or translation, chalo we talk about translation only, that translation process between bacteria and animal shows a lot of similarities. That means the three processes are almost the same. Initiation, elongation, and termination. These three processes are almost the same. There are some other factors that are there in eukaryotic system. Eukaryotic system is a little more complex because you have some additional ribosome, additional units, and little more transcription factors are there. But the basic mechanism of initiation, elongation, and termination, it remains the same. Not only the basic mechanism of initiation, elongation, termination remains the same, but the codons that codes for different amino acids, that is also the same only. So AUG codes for methionine in bacteria also, AUG codes for methionine in animals, in eukaryotes also, in animals also. So through evolutionary studies, it has been found that bacterial mRNA codons are more similar to animal codons and not to virus codons, plant codons, or mitochondrial codons are not relevant here. Bacterial codons are similar to animal codons. Is it clear?
Okay, ma'am. Thank you, madam.
Okay. So now next question is, all mutations are detected and eliminated from cells. True or false? All mutations are detected and eliminated from cells. Let's see the answers. False. So Kanchana has said false and Janara has said true. Okay. The correct answer is false. Now why? Now, can you tell me the basic reason of cancer? There are mutations, right? The fact that cancer happens to human body The fact that cancer has become so prevalent and so common, unfortunately, today is because so many different mutations are happening in our body. Because we are exposed to so many different kinds of oncogenic materials, so many radiations, so many cancer causing agents that we are unknowingly eating and we are exposed to. So, although our body tries its best to eradicate the mutations, but still there are so many different kinds of mutation that do exist in our body which are not eliminated, that ultimately leads to cancer progression. So the fact that cancer happens to human body itself shows and says that all mutations are not eliminated from the body. They can be detected, but they might or they might not get eliminated. Okay? So the correct answer is false. Then low mental and physical stress maintain long telomeres. True or false? This we have studied in last week also. Think. Okay. The answer is Kanchana has answered D true, Jenna has answered true, yes. So obviously if you have low stress, then stress is the cause of so many problems. So the answer is true. Next question is last question. Again, this we have studied in the last week also, that adequate vitamin C in diet ensure longer telomere length. True or false? So Kanchana has answered false. Other responses? Okay, fine. Sorry. The correct answer is true. So, if you remember what we have studied in the last class, we have studied that vitamin C in our diet, vitamin C has a lot of antioxidants. So, just see if stress is there in the body, stress produces a lot of oxidative reactive species in our body. Why stress is so problematic for our body? Because stress produces a lot of reactive oxidative species. Stress produces ROS, and this reactive oxidative species can be removed with the help of intake of vitamin C, because vitamin C has a lot of natural antioxidants. Therefore, if we have adequate amount of vitamin C, then it will ensure longer telomere length. Okay, so today we have studied about epitranscriptomes, means that different kinds of RNA modifications that takes place, and different kinds of RNA modifications can be regulated by writers, erasers, and readers. Writer, it writes the modification, eraser erases the modification, readers reads the modifications. Then we studied about protein synthesis machinery, translation, about the ribosomes, about the genetic code, about how to read the triplet code, what are the charging of the tRNA, and then the structure of the tRNA, then the codon anticodon base pairing we have studied, ribosome composition, and then the initiation, elongation, termination we have tried to touch it. We could not complete it. If you have any doubts in that, we will I will explain you in the next class, I mean in the last class, that I think I will happen on Tuesday. Okay, so now I'll just end today's class. We'll meet again on Tuesday. Fine. Bye everybody.